particles in an electric field with the help of electric field. So that the technique is called as electrophoresis. It is a technique of separation of macromolecules like DNA, RNA and proteins. The term electrophoresis was derived from a Greek word phoresis, which means to get carried away. So then why it is called as gel electrophoresis? Because gel is used as a medium to separate fragments of this DNA, RNA and proteins in the electric field. So that's why gel is added before the electrophoresis. Electrophoresis is the process, is the technique, whereas gel acts as a medium. We have different gels. So we have two gels, agarose gel and polyacrylamide gel. Now let us first see about agarose gel electrophoresis. So agarose gel electrophoresis, in this, its pore size is large and large macromolecules like DNA and other nucleic acids and protein complexes are separated in this agarose gel electrophoresis technique. So now methodology of agarose gel electrophoresis technique. First we have to prepare the gel. For that we have to take agarose powder. We have to take agarose powder and we have to add it to the electrophoresis buffer. And the concentration of agarose solution depends upon the size of the fragment. If the size of the fragment is large, then the concentration of agarose solution will be low. If the size of the fragment is small, then the concentration of agarose solution will be high. So after mixing agarose powder with electrophoresis buffer, we have to add ethylene bromide at 0.5 mg concentration. For visualization of DNA fragments after electrophoresis, we have to heat in microwave. After heating that, we have to cool down in 60 degrees Celsius. Then our agarose solution is prepared. Now we have to take a cashew tray with a glass plate. In this we have to place the comb. We have to place the comb before adding agarose solution, because if we directly add agarose solution and later it gets solidified and forms as gel. After formation of gel, if we place the comb, that might not give the accurate results. So it is suggested that to place the comb before we pour the agarose solution. So now after placing this comb, we have to add agarose solution, which is heated before. So now it is allowed to solidify at room temperature. After that, we have to remove the comb very carefully without breaking the walls. Walls are formed.
They are not walls, they are wells.
Okay. Wells are formed. Agarose wells are formed. Now in this wells we have to add DNA fragment solution along with electrophoresis buffer with the help of pipetting. Now after adding, we have to introduce this into electrophoresis chamber. And we have to let the electric current pass into it. Then bubble formation takes place in these balls, and as DNA is negatively charged, it moves towards the positive electrode, which is red in color. So the DNA migration can be started by using dyes such as bromophenol blue, and xylene siler. So this is about agarose gel electrophoresis. Now let us go with page. Page is polyacrylamide gel electrophoresis. SDS page. Now it's for SDS page. Sample. SDS is sodium chloride sulfate. So we have to take protein and SDS, sodium chloride sulfate. We have to add a reducing agent, which is beta pentactoethanol. Now what happens is we add beta pentactoethanol into this protein and SDS solution to give a denatured. Denatured means the disulfide bonds are broken. That means beta pentactoethanol denatures this solution and breaks disulfide bonds. So SDS state is prepared. Now we have to do gel casting. Further we have to take vertical electrophoresis, vertical casting way. In this we have to pour the solution. We will get two types of gels, stacking gel and resolving gel. So stacking gel, as the name says, stack means it binds the proteins as bands. Now resolving, it separates the protein fragments according to their size. So this is gel casting. Now we have to load the gel. Same like agarose, how we did. In the same way we have to load the gel with the help of a micropipette. So after loading, same electric current is introduced into this, and the proteins move towards the positive electrode, which is anode, and we can visualize this activity with the help of dyes such as silver stain, fluorescent dyes, and brilliant blue. Now for analysis we can use molecular weight markers and densitometry.